All right, innate defenses against infection. So we're going to talk about the innate or non-specific side of the immune system. There are lots of different defenses our human immune systems have against pathogens. They can be physical, chemical, cellular. They can involve pathogen recognition and phagocytosis. They can cause inflammation and fever. Okay? So our innate responses are really the first line of defense against pathogens. The key is that it's nonspecific, right? Cells in our innate immune system, which we'll talk about in a minute, are widely spread throughout the body and throughout tissue. And so as soon as a pathogen enters, essentially, they should be able to recognize it and either take it up and destroy it or alert the immune system that something is going on. And by doing so, they can help prevent infections from starting in the body. Innate immunity is built in. It's not acquired over time, right? We always have these cells, okay? But there's no memory, so they don't have an improved response against pathogens that they see repeatedly, unlike the antigen-specific side, which we'll talk about next time. But they do provide an immediate and rapid response, which is so important in stopping an infection from starting. But some pathogens have evolved ways to get around them. And sometimes the innate immune response can damage the host tissue. Let's talk about the physical defense first. All right, so not always considered part of our immune system, but the human body has some really, really good, strong physical barriers in place to prevent infection. Okay, at the most simple level, our cells are joined tightly together with tight junctions, right? Pathogens can target the extracellular proteins and matrix that hold the cells together, but when it's intact, they have a really hard time gaining access to tissue. Here are some pictures of junctions here. We're not really going to get into it. I'm assuming you guys cover this in A&P anyway, but there are different kinds of junctions. The key here is that in order to get into tissue, pathogens have to get through the extracellular matrix holding the cells together and through those junctions. Our skin itself is the probably most effective barrier against infection that we have. And there are lots of reasons for that, right? We know that we have 3 different layers, right? The epidermis, the dermis, and the hypodermis. The cells are packed very tightly together. This makes the skin tough and resistant to degradation, right? On the surface of the skin, we have fatty acids that create kind of a dry environment, a slightly salty environment, or slightly acidic, and all of those things can inhibit the growth of bacteria. And on the surface of our skin, we have dead cells, right? Dead skin cells that are shed regularly. And when those cells are shed, they take the microbes with them that were on the cells. So that helps kind of keep the numbers of bacteria on the skin down. But when the skin is broken, infection can occur, and it can occur pretty easily. Other physical defenses are mucous membranes, right? Our mucous membranes are a huge portal of entry for pathogens, but they do have some physical defenses that try to keep pathogens out. Okay, again, here you have tight junctions. You have epithelial cells packed closely together. You have mucus, right? Mucus is actually really beneficial in keeping pathogens out. It protects the cells underneath it. It can trap debris, particulate matter, including microbes, and kind of flush them away from the mucosa. It can contain different antimicrobial peptides, and it gets flushed out of the body regularly. So when it gets flushed out, it takes the bacteria with it, kind of like the dead skin cells do. We've got mucus in our respiratory tract, in our digestive tract, that got cut off, but our genitourinary tract, pretty much anywhere that there's a mucous membrane, right? Hence the term. Other defenses, shedding of skin cells we talked about, expulsion of mucus we talked about, excreting your waste, right? You are excreting a huge amount of bacteria with that. The flushing action of urine and tears. So urine actually flushes the urinary tract on the way out to keep pathogens out. Your tears help keep pathogens out of your eyes. In terms of your eyes, you also have your eyelashes and your eyelids that help keep things out. And all of those things can carry microbes away from the body. Touch lightly here on the microbiome. We've already talked a lot about microbiome, right? It is actually helpful and in and of itself is a defense against pathogens because it competes with pathogens for binding sites and nutrients, right? Space and nutrients. If the good bacteria are there, then there's no space or nutrients for the bad bacteria to take up, right? When the normal microbiome is disrupted, then you have less good bacteria, you have an increased amount of space and nutrients available for pathogens, right? Like C. diff in the gut or Candida albicans in the female reproductive tract. All right, chemical defenses. So our body produces a large array of different chemical mediators for infection and for the immune response. They're found in various tissues and body fluids. There are lots of cell or chemical mediators that can work together, and we'll talk about those in a little bit. Some are produced endogenously by our own human body cells, and some of them are produced exogenously by cells not of our body, which are essentially cells of our microbiome, right? Some chemicals that help keep bad bacteria away are made by our good bacteria, and some of them are made by our cells. So know the difference between endogenous and exogenous. Some chemical mediators are produced all the time, and some of them are only produced as needed. A couple of really important enzymes. Lysozyme. Lysozyme is in our spit, it's in our tears, it's in a lot of different places. It breaks down peptidoglycan. And as we've come to learn about bacteria, they all have peptidoglycan, right? Lactoferrin, we talked a little bit about iron sequestering when we talked about pathogenicity. Some bacteria will try to take the host's iron away from the host and use it for the bacteria. Well, we make a substance called lactoferrin that helps stop that from happening. In our skin, our sebaceous glands make sebum, right? Sebum actually helps seal off the pore of the hair follicle to prevent bacteria from getting in the first place. However, some bacteria can feed on that. The good news is that some of the good bacteria in our skin can feed on that and colonize that and secrete chemicals that can inhibit other microbes. Some of them lower the pH of the skin. We know that bacteria don't like it very acidic, right? If the humidity is low or your sebum production is decreased, then that reduces the amount of good bacteria there on the skin, and it makes the skin more susceptible to infection. Right, in the GI tract, we have lots of defenses in our GI tract against GI pathogens. Our saliva has many different enzymes, including lysozyme, to break down pathogens. We've got mucus in our esophagus. Your stomach acid does a very good job of killing a lot of things that come into contact with it because the pH is so low. You also have enzymes made in your intestines, antibacterial peptides made in your gut. You have IgA, immunoglobulin in your gut. That's made by B cells, which we'll talk about in the next lecture. Your liver bile is antimicrobial. There's lots of things in the gut that try to decrease the amount of bad bacteria that can be there. Our urinary tract, because of that constant flushing of urination, not a lot of bacteria get in, and when they do, they cause an infection. A lot of times, urine is also slightly acidic, which can help inhibit pathogen growth as well. The female reproductive system has a host of different mediators to help prevent infection because we really don't want an infection in the female reproductive system, right, in order to successfully reproduce. One example is the lactate produced by the lactobacillus bacteria in the microbiome. It helps keep the pH low, inhibiting pathogens. We talked about our tears already. Our tears contain lysozyme and some other enzymes to help keep pathogens out of the eyes. Earwax is actually good for your ears. It contains fatty acids that lower the pH and help prevent bacteria from getting in the ears. Respiratory tract, we've got lysozyme, lactoferrin, other enzymes in the mucus itself. And then there's the physical mediators as well, like the cilia and things that help get things out of the lungs and the airway. In humans, we have a number of antimicrobial peptides. A peptide is essentially a short piece of protein that's too short to be an actual protein. So it's a short chain of amino acids that are also antimicrobial in this case because they're antimicrobial peptides. A lot of them are induced when a pathogen's presence is sensed, and some of them are made at low levels all the time in our cells. They can damage cell membranes, DNA or RNA of a pathogen, interfere with cell wall synthesis or making that peptidoglycan. A great example are the defensins produced by our epithelial cells and some immune cells. They're secreted to damage the plasma membrane of a pathogen. Right? Bacteriocins are produced by the good bacteria in your gut that help prevent other gut-dwelling bacteria, aka pathogenic gut bacteria, from taking over. Another concept that we'll talk about here, but we'll talk also about in the next lecture, is opsonization. So when we talked about pathogenicity, we talked about how adhesins on the surface of a pathogen need to bind to ligands on the surface of a host cell, right? That binding has to happen for pathogens to get inside cells, right? And you can block that by blocking the adhesins or the ligands. Okay. So there are some proteins made by our immune system that can actually kind of block the adhesins on the surface of the pathogen so that it can't get into the host cell and it can't cause any damage, right? So it effectively like inactivates the pathogen. Okay, and when the entire surface of the pathogen is covered in those proteins, that's called opsonization. A lot of times, those proteins that are opsonizing a pathogen will also signal to the immune cells, hey, come gobble this up and get rid of it. Our plasma has a number of protein mediators in it that help get rid of pathogens because we really don't want pathogens in our blood, right? Our liver makes some of them, like C-reactive protein, ferritin, transferrin, fibrinogen, mannose-binding lectin. Those are just examples of some of the proteins in your plasma. We also have this really interesting system of proteins called complement. Complement proteins are a system of more than 30 proteins, where we keep finding more new ones, that help the innate immune response. They help communicate between the innate and adaptive immune responses as well. So complement is a really complicated system. And you don't have to know a lot about it, but these are proteins that kind of normally circulate in the blood as inactive precursor proteins. They don't become activated until there's an immune response. Some of them can attach directly to pathogens. Some of them can bind to antibodies that have bound to pathogens. And they essentially can form complexes that poke holes in the membranes of cells, like gram-negative bacteria, killing them, right? Because we want to kill the pathogens. The other chemical mediators that we make are cytokines, right? We've talked a little bit about cytokines. Cytokines are different proteins that communicate from cell to cell and in the immune system. A lot of times cytokines are produced by cells when they sense an infection. Those cytokines will travel through the body and bind to receptors on whatever cell they're going to bind to, right? And that triggers even more immune response. And so that effect can cascade and create more immune response, and more immune response, and more immune response, and really activate the immune system. It can also overactivate the immune system if there's too much of it. When the immune system is activated by cytokines, it can cause things like activating your B and T cells. It can stop cells from dividing if they are infected. It can cause apoptosis in cells, which is a really cool thing that our cells can do. It's essentially programmed cell death is how we refer to it. Essentially, the cell dismantles itself from the inside out. Apoptosis can be good because when a cell undergoes apoptosis, if it's infected or damaged or something, then it won't necrode. It won't cause necrosis. It'll just kind of break up into little pieces and float away. Or it can cause chemotaxis. So chemotaxis is essentially the trafficking of cells, like our immune cells, to areas of infection in the body, right? They have to know where to go. And in order to know where to go, they have to have some sort of chemical signal. And that chemical signal can be from cytokines and chemokines. Some of our cytokines are called interleukins. There are a number of them. They are specific proteins made by immune cells to signal to other immune cells. Sometimes they can even be recognized by other host cells as well. They usually have numbers like interleukin 2, interleukin 7, interleukin 17. Interleukin 2 is really well known for kind of beefing up the immune response and activating adaptive immune cells like T cells and B cells. Chemokines, like I mentioned briefly a couple of minutes ago, are chemotactic. They cause the recruitment of leukocytes to sites of infection. Leukocytes are your white blood cells, right? So chemokines are produced at sites of infection. They're also produced at sites where tissue is damaged, so wounds. They can recruit your white blood cells to wounds, and some of your white blood cells actually function in wound repair, which we're not going to get into in this class, but it's actually really cool if you're interested in learning about it. And some of them can cause just general inflammation. And some complement proteins and interleukins are also chemokines. So it's this whole big web of things. Okay, chemokines are any kind of protein that can communicate from one cell to another, right? Interleukins are cytokines that signal from one immune cell to another for the most part, but can be recognized by other cells. Chemokines cause the movement of cells to sites where they are needed, right? Another kind are interferons. Interferons are immune signaling molecules that are important against viral infections. Remember, I said that when cells are infected with virus, sometimes they'll make interferons that can signal to other nearby cells to protect themselves against infection. Other kinds of interferons are also made by our immune cells, and they help to kind of ramp up the immune response against viral infections as well. All right, let's talk about cellular defenses. In the blood, all of our blood cells are formed by a process called hematopoiesis. Right, it happens in the bone marrow. It comes, all of our blood cells come from hematopoietic stem cells. Our red blood cells they're called erythrocytes. They carry oxygen to tissue, right? Our platelets or thrombocytes help form clots and tissue repair. Our white blood cells, our leukocytes, are what are our innate and adaptive immune responses. And in this chapter, we're going to focus on the innate side. And the next chapter, we'll focus on the adaptive side, okay? So in general, our white blood cells can be granulocytes or agranulocytes. So granules are just little pieces of chemicals stuck together. They make little granules. They look grainy if you're looking at cells under the microscope. So when we're talking about our innate granulocytes, we can differentiate them based on appearance of nuclei and appearance of granules. So we've got neutrophils eosinophils, basophils. Neutrophils eliminate or destroy extracellular bacteria. They migrate through blood vessels to areas of infection and tissue damage. They can seek out and kill infectious bacteria. They can then ingest them and kill them, or they can secrete granules and kill them. Right? When a cell secretes granules, it usually secretes a large amount of granules at once. It's called degranulation. It spits out all the granules. And those granules are essentially proteins and other chemicals that can kill pathogens. Sometimes they can create like this kind of mesh network to kind of trap pathogens, making them easier to gobble up and destroy. However, sometimes it can kill host cells as well as pathogens. When you have a buildup at a site of infection of neutrophils and dead neutrophils and cell debris and bacteria and dead bacteria, that is collectively called pus. And then we have our eosinophils. Eosinophils are important for protection against protozoa and helminths, but they also play a large role in allergic reactions. Their granules contain histamine. They also contain degradative enzymes and a protein called major basic protein that actually can bind to the surface of parasites and disrupt the host cell membrane, disrupt the parasite cell membrane. Another granulocyte are the basophils. Basophils are very important in inflammation and in allergic responses. Basophils can be triggered to degranulate their granules by complement proteins such as the aflatoxins. Okay. Basophils also contain histamine in their granules. They can also help to open the gaps between cells so that inflammation can occur. So rapid influx of fluid and immune cells into tissue or areas of infection. Mast cells are similar to basophils. They reside in tissue for the most part. They leave the circulating blood and go out into the tissue. They can induce an inflammatory response when infection is detected in tissue itself. And the opposite of granulocytes are agranulocytes. They lack visual granules. For the most part, agranulocytes are lymphocytes or monocytes. Now, lymphocytes are your natural killer cells, B cells and T cells, which are the adaptive immunity. Your monocytes are precursors to macrophages and dendritic cells. Macrophages and dendritic cells are two very important parts of your innate immune system. First, a little bit about natural killer cells. They are mononuclear lymphocytes. That means when you're looking at them, you only see one nucleus. When you look at neutrophils and some of the other granulocytes, their nuclei look very unusual. Natural killer cells recognize and destroy cells that are abnormal, including cancer cells and cells infected with virus. Recognition involves identifying very specific molecules on the cell surface called major histocompatibility complexes. Major histocompatibility complexes, or MHCs, are a group of proteins on the surface of a cell that display little chunks of protein or peptides of things inside the cell. Usually there are proteins being synthesized inside the cell. And if those proteins are normal and normal self proteins, then NK cells and other cells won't be triggered. If they're abnormal or if they belong to a pathogen, then NK cells or T cells, which we'll talk about next time, can recognize that as bad and kill it. So when an NK cell recognizes something that's bad, it can trigger that cell to undergo apoptosis, or it can release this protein called perforin that pokes holes in the cell membrane and kills the cells. Monocytes themselves have a nucleus that lacks lobes and no granules. Okay, so different again than neutrophils and things like that. They're phagocytic. These guys will engulf particles. That can include pathogens, that can include cells undergoing apoptosis to break them down and dismantle them. They're very important in the recycling of cell pieces, and the breakdown of pathogens. Monocytes circulate around in the bloodstream. When they leave the bloodstream, they differentiate either into macrophages or dendritic cells. Macrophages and dendritic cells can live in body tissue for long periods of time. They will live in that body tissue and kind of survey the environment regularly to see if there are any pathogens present. If they encounter a pathogen, they will produce cytokines and they'll gobble up that pathogen. And a lot of times they'll take that pathogen, if they're a dendritic cell, they'll take that pathogen back to the lymphatic system and to the T cells and B cells and tell them, hey, look what I found. This is here, you need to activate against it. Macrophages macrophages are unique because they can develop differently depending on the tissue that they're in. So macrophages become the microglial cells, the Kupffer cells of the liver, things like that. Dendritic cells typically reside in the skin and mucous membranes. They encounter a lot of pathogens and other particles, and they're really key in alerting the immune system to something gone wrong. Let's talk about pathogen recognition. So we already talked about opsonization, right? When a pathogen is coated with either chemicals or antibodies made by your B cells, they can be recognized by the innate immune system and phagocytosed and broken down. But phagocytes can also recognize other structures that are common on the surface of many microbes. They're called PAMPs, or pathogen-associated molecular proteins. Things that many pathogens have, like peptidoglycan, flagellin on the flagella of pathogenic bacteria. It's a little bit different than the flagella on human cells. Lipopolysaccharide, other lipopeptides expressed by a lot of bacteria. Bacterial DNA and RNA looks a little bit different because of the chemicals associated with it than human DNA and RNA. So all of those things can be patterns that phagocytes can recognize. When one of those PAMPs binds to a receptor on a phagocyte, it triggers the phagocytosis process to happen. So it triggers the cell to extend its cell wall and plasma membrane out around the pathogen and take it in. Once the pathogen is brought in, the phagocyte forms a vesicle around the pathogen. So on the way in, essentially, it forms a vesicle because it has a plasma membrane all the way around it. And then once inside the cell, it actually fuses with the lysosomes inside the cell, which contain really strong, powerful digestive enzymes. And when that fusion happens, it's called the phagolysosome, and those digestive enzymes do the work on the pathogen that got brought in and break it down into lots of little pieces. And then all of that waste gets excreted from the cell. Let's talk a little bit about inflammation and fever. So we talked about fever a little bit already. Inflammation is the inflammatory response triggered by a cascade of chemical mediators and cellular responses that occur when cells are damaged or stressed or when pathogens invade. That is the definition of inflammation. Very broad, right? Lots of things can trigger inflammation. Pathogens can trigger inflammation. Cellular stress can trigger inflammation. Damage to cells can cause inflammation, lots of things, right? Inflammation can be good because it helps recruit your white blood cells to areas where they're needed. And those white blood cells can eliminate pathogens, they can remove dead or damaged cells, or they can start repairing damaged tissue. Remember, some of your white blood cells actually function in wound repair. However, excess damage to local tissue can happen, and it can become deadly for too much inflammation. Acute inflammation are those quick, early responses to tissue injury, right? In acute inflammation, you have blood vessels that constrict really briefly to minimize blood loss, and then they dilate and increase their permeability so that immune cells can get out of the blood vessels into tissue. Okay, when this happens, this is when you get redness, swelling, heat, pain, which can further enhance the inflammatory response. This increases the neutrophils in the sites of infection, plus lots of extra fluid. So you get lots of pus. After a few days, your macrophages will actually start clearing out the pus. And eventually tissue repair will begin. Chronic inflammation is a lot different than acute inflammation. Chronic inflammation happens when acute inflammation doesn't clear all the way. A lot of times that's in response to a viral pathogen or a bacterial pathogen, or an ongoing low-level battle between the host and pathogen. So a pathogen that's there, not doing a ton of damage, the immune system's recognizing it, but not ramping up a ton. So there's this low-level inflammation that happens. Chronic inflammation can help a wound, or chronic inflammation can also occur when a wound heals on the surface, but maybe not deep down inside all the way. So because it's not healed all the way, the body is still making some inflammation to try to get those repair cells there. Chronic inflammation is involved in a lot of diseases. You guys are probably, you've heard of it before, right? Inflammation is bad, right? In your joints, in your tissues. Chronic inflammation can be involved in Alzheimer's and Parkinson's, heart disease, certain cancers. It can cause the formation of granulomas. If this happens in the lung, they're called tubercular granulomas, aka tuberculosis, where immune cells keep trying to kill the pathogen, but the pathogen has isolated itself inside the granuloma and it's unreachable. This can cause a lot of scarring, like cirrhosis of the liver from Hep C. We already know a little bit about fever, right? Fever gets induced in response to cytokines made in response to pathogens, right? And those cytokines are called pyrogens. They are produced during infection. They can be endogenous or exogenous, okay? So we can make them ourselves, or other microbes can make them, right? Lipopolysaccharide itself is a pyrogen. So the presence of lipopolysaccharide can cause fever, right? Fever in moderation can be good because it stimulates your white blood cells to kill pathogens. And it can inhibit pathogen growth a little bit because you're raising the temperature a little bit above the optimum temperature for those microbes to live. It can cause the constriction of your blood vessels, which can cause skin to appear pale because it's diverting blood flow from the extremities, right? It's thinking, preserve the core, right? Shivering helps to generate heat. Sweating can help cool the skin. There's lots of things that can happen during a fever. Sometimes, though, that immune response is too big, right? We've talked about superantigens, overactivation of the immune system. Overactive immune cells will just start killing everything they find. And that causes tissue damage, organ damage, life-threatening fever, and death. And that is the end of innate immunity. Next time we'll talk about adaptive immunity.